I would like to know really from our physicians, what is your experience which you have on these combo products, or what do you know about it? I think we don't have really combos in the market itself, but you might use these combinations for treatment in your patients. At the beginning, there was, I remember there were big efforts, even Novartis, they had a program of 6 different products they wanted to combine with Veruxo, and they suddenly stopped it, I assume for reasons that they thought it was not so interesting. But it went on, and what I heard last year in ASH, that in general physicians were not very happy with the results of these combos. Now it looks like it's coming up again, so we're a little bit confused. Maybe you can explain us a little bit more what is the today's situation of combinations. Whoever wants to start. I mean, Ruben has left, so we don't have to wait for him.
Okay, so I'll just maybe say 1 or 2 introductory words. The concept is to use drugs that work in different ways, so that you can target concurrently more than one pathway within the malignant stem cell, and in that way try and be more effective in killing the cell, stopping its proliferation, stopping its interactions with the microenvironment around it, and just in overall have more effective treatment. The concept is not unique. It's a concept that exists in oncology, it exists in hematology, where combination therapy has become very popular and very successful. In fact, it's even in unpopular and unsuccessful ancient chemotherapies, for example, in acute myeloid leukemia, combination therapy has existed for 4 decades, 5. I don't know when they started using chemotherapy for AML, probably in the 70s. And so monotherapy in cancer has never really been highly successful, even with the older drugs, with the chemotherapy drugs. And now with the targeted agents, the combinations are back in. Lymphoma, multiple myeloma, Hodgkin's lymphoma, you name it. Almost every disease where there is more than one active drug, people have combined the active drugs together, which work differently. Sometimes the advantage of using the drugs together is that they'll have different efficacy in killing the cells that you want to kill. And sometimes the advantage is that they will have a different side effect profile from one another. So you can use different drugs that don't have the same side effects and not get a cumulative effect in a negative sense of using the same drugs, so the drugs with the same type of side effect in the same patient, where the toxicity of the drug becomes prohibitive. So there are a number of combinations that are in clinical trials. It should just be said that all combination therapy in myeloproliferative neoplasms is investigational. So there are no combinations that are licensed. There are no combinations that are used in routine clinical practice. The combinations we started using them in relapsed or refractory disease, and now the combinations have been moved up into frontline treatment, particularly in myelofibrosis, but in other diseases as well. And perhaps I'll let colleagues expand on specific examples. That's okay.
Before you continue, could I ask you to sit here? Maybe it's better, because otherwise people turn around his head, and at the end we will have some hospitals. Thank you very much. It's much better here. Yeah.
So maybe if we're going to get into specifics, maybe I'll start with combinations in PV, which are less, and then give you the MF combinations, or I can start with some. So most of the combinations are being studied in myelofibrosis, which is more advanced form of MPNs. And I think just to make the point of combinations, like what Martin just said, is that it's important in cancers we use combinations, pretty much in all malignancies. I think the idea of single agent therapies in MPN started with CML, where one typically monotherapy, so one drug is effective, but this is a different disease, and I think we have to treat it similarly to the other hematologic malignancies. So I start with polycythemia vera. So in terms of combinations, one of the combination that is being used, I think, somewhat off-label, is a combination of interferon and ruxolitinib. They do have some overlap in toxicity, so they will both affect blood counts and can result in lowering of the blood count, so would need to be watched really carefully. But I think there is some positive data on the combination of those two drugs, where some of the side effects that are seen with interferon, for example, such as fatigue, can be opposed by ruxolitinib in a way. And just the mechanism of action of the two drugs could potentially be complementary. I mean, the truth is that there are many different pathways that interferon affects, and we don't really know exactly how it works. But it does seem that the combination of the two drugs can be effective, but it really needs to be done in specific population of patients. So it's not a combination that should be used for everyone. And I think studies are ongoing, and we'll have more results to see which patients are the best candidates for this combination. The second combination in PV, and that's purely investigational now, is rusfertide, for example. So this is a hepcidin mimetic that is being studied in PV. And the reason why I'm saying it's a combination is because it's being used as an add-on therapy. So for those patients that are just being treated with phlebotomies alone, it's combined with phlebotomies. But for those patients that are being treated with other cytoreductive agents such as hydroxyurea, ruxolitinib, or interferon, it's added on to that therapy for those patients that are heavily phlebotomy dependent. And the advantage of that is that rusfertide controls the need for phlebotomy, so it controls the hematocrit. And the idea is that if the hematocrit is kept under continuous control, then presumably it results in better control of the disease, as well as potentially better control of the symptoms that may be specifically related to iron deficiency. So those are the two combinations in PV that I think are the main combinations being studied. Now, in myelofibrosis, many, many more combinations, and I don't know if we're going to go through all of them. So I can start with the one that I am most familiar with, is pelabresib combination. So the study's been ongoing for a long time. And initially, we start these combinations in sort of second, third line setting of the disease. So there's two ways to approach it. One is patients that are already on a JAK inhibitor, so on a ruxolitinib, for example, and then maybe they're losing their response or they're not having suboptimal response, the drug is being added on. So this is, we call it relapsed refractory setting or a second line setting. And so you get additional benefit from addition of the drug, which is a class of drug called BET inhibitors. So it's affecting the chromatin, which is basically the packing of the DNA in the malignant cells. And so the two drugs together, you get additional benefit from what you can see with each of these drugs alone. And the positive results in the second line combination resulted in moving that combo up front. So for newly diagnosed or recently diagnosed myelofibrosis patients who have not been treated with with a JAK inhibitor combination up front, so you start immediately on the combination. And so the early phase 3 trials were presented at ASH, showing that there is significantly improved significantly improved spleen responses in the combination of pelabresib and ruxolitinib as opposed to ruxolitinib and placebo. And there is some improvement in symptoms as well. And I think it's important to point out that symptoms are difficult because ruxolitinib is actually, especially in patients that are relatively newly diagnosed, ruxolitinib is very effective in controlling symptoms in a lot of patients. And so to be able to show additional benefit that is statistically significant is actually not an easy target, I think, to show. But there was an improvement in symptoms as well. So I think so. These are the one of the combinations that's being studied right now. The study is ongoing, and what will be really important to see is that are these responses durable? So are we going to continue to see that patients who are getting the combination up front expected to do better than those patients that first get ruxolitinib and then get pelabresib perhaps added on? And those questions are more complicated, but as these studies go on for longer periods of time, we'll be able to get some of these answers. So I'll give it to you for the next one.
Perfect. Thank you very much. So for first thing to basically look at the combination to me is to answer the question, who does need a combination? It's certainly not every patient. At what point to use the combination? As Dr. Kremanskaya said, is it at the beginning? Is it at the time we believe that the single agent is not providing benefit? How to define the non-providing benefit? So who is considered not ideal responder? And then I think the most important is, what are we trading in? Are the combinational adverse events acceptable compared to what we can gain, or is it too toxic? So as she described the study with the bromodomains inhibitor, which are actually molecules that work on the cell levels, improving the additional inflammatory pathways to shut down, which the JAK inhibitor is unable to do. And that is a concept across the whole MPN or myelofibrosis, because the disease comes with a lot of inflammation, that even despite the JAK inhibitors, which at this level the only one that has been studied in combination has been Jakafi or the ruxolitinib. Now we're looking more into combining other JAK inhibitors with new molecules. So that is going to be the new era, because some of them, particularly latest approved momelotinib, are not myelosuppressive, which has been actually the biggest side effect that we've seen on the second combination, which was the JAK inhibitor and something called BCL-XL, which is a molecule that works as well in the inflammatory pathway and as well as pushing the cancer cells to die. They're kind of resistant. Cancer cells don't want to go away and die. So we have a group of agents that are working on that pathway to basically just trick them and shut them down, tell them, hey, you're going to go and disappear. So that's what we combine with called the navitoclax. And that has been also very successfully studied, shown to provide benefit in controlling the spleen. It was actually over 60% of patients, which with a single agent seen only in about 40% plus or minus. But the symptoms were comparable between two arms, and the side effects profile was concerning. There was significant, what we call cytopenias or bone marrow suppressions. So that's something where we have to look at how much are we trading in. I think it has a great concept for adding on, so that drug, which is probably not going to be developed anymore because of these results, but could have added a lot to patients. They are not doing well on the monotherapy and then being added on with a reasonable approach. Frontline, it is myelosuppressive. So it is putting too much risk for newly diagnosed patient for an unknown benefit in terms of the longevity. So we don't know how much would we be giving in for people to respond. The biggest concept of that combination is to prevent the disease to progress or stop the progression that could happen ultimately at some point. So kind of like changed the disease course down the road. If that's something we'll be gaining, then we'll be able to basically sacrifice some of the side effects if they're manageable, because we'll be changing the disease from progressing to leukemias or somewhere else. There will be definitely a gain point. But if we are not showing that, we only showing we're gonna just improve the spleno symptoms for, let's say, a defined period of time of 1 or 2 years, then that side effects with that may be very concerning. So that is something really important to realize when we are talking about combination, to really pick the right situations, the right patients, and the right drugs. Then we had a couple other very interesting studies. Something that is ongoing currently has also combining the JAK inhibitor ruxolitinib and a molecule called export inhibitor, or selinexor, which is on a different level of entering the cancer cells as well as shutting down the extra pathways. That is currently enrolling in a frontline setting. The other drug is only taken once a week because selinexor has already some history from other cancer, especially acute leukemias, and it was quite toxic. Significant nausea, diarrhea, and intolerance. So the once a week concept with taking mandatory 2 extra drugs to prevent the nausea, it's something a little challenging, but if we are able to show that that is going to change the course of the disease and nobody's going to progress to leukemia, that will be the win. So at that situation, we kind of always have to weigh in what is the gain, what is the risk, and look at the bigger picture and really choose the right patients, not to do any harm, but actually help where the help is needed. So therefore, sometimes these frontline approaches and the combination when we don't really understand could be very challenging, and we could not intentionally get rid of a good drug because the design of the study or the results were not expected as we would expect. But that was just because we just basically wanted to explore it rather than, hey, is it the right patient population, the right design, and is it the right endpoints we're looking at that we could add on the combos? So that is something that we work on, but that's always considered in when people come in and ask, hey, why am I not the best patient for this particular clinical trial? That's exactly what we're looking at. Like, your disease may be too good or it's too bad. We don't know what it's going to do, and so on and so forth. So that comes with that. At the Anderson, at our institution, we do lots of these investigational initiated studies where they're not part of like a global US development, but they come either from our lab or from a small collaboration with the companies. So we basically explore the efficacy in 20, 30 patients first to see whether that's even a signal, and then develop the collaboration with other companies on the global scale, or move it even to other countries to basically, hey, that this is it, this is something that's reasonable. But lots of those then are early phase studies in a combination where we're looking at the safety first. So once we breach the safety, we say, hey, this is perfectly to combine. We can move on and look at the efficacy, and then we have to design it the best way possible. Like one that's coming now, it's going to be something called MDM2 inhibitor, which is another molecule working in the cells being pushed into dying. And it's by a pharma company that is now doing a phase 3 study adding on the MDM2 inhibitor to people with suboptimal response to Jakafi. But that study will have people starting on Jakafi and with very exact assessment at certain standpoint, and then once precise criteria are defined, these are the patients, they are not responding at a certain time point, then they will be randomized to either continue the drug or adding on the new compound. So as Dr. Kremanskaya rightly mentioned, it's going to help us to assess what is the gain benefit on top of the other drug. Because so far, a lot of these studies had kind of not really well-defined criteria where we had mixed up all different suboptimal non-responders or responders, where some people were intolerant, some people couldn't take the medicine because of side effects, some people had a large spleen still, some people had significant anemia, so those were a little bit more of a mixed population. So now we hopefully gonna learn more how to best design this and what we can gain from it. At our place, we also trying to add on another compounds. For example, we had an approach, and you're gonna be joining it soon, with the Tregs, which is an immunotherapy add-on to Jakafi, which are basically cord blood-derived cells. They've been extracted from the cord blood, they purchased standardly from the blood banks, and then the T regulatory lymphocytes are being basically engineered and then infused to patients, because those are the cells they are lacking in the myelofibrosis patients, in an intention to educate the immune system. And that's going to go in people suboptimal to ruxolitinib, and it comes along with a lot of blood work and a lot of regular dynamic assessments to really starting to understand, as we said, why are people not responding ideally, and what are the molecules or the markers they are responsible for not ideal assessment. So we'll be more tailoring the treatments to what is actually behind it and what can we do about that or no. And I'm hoping that kind of a lot of this data will help us to develop other combinations for the future. So we will really understand where we have to do them, how much benefit are we adding, and is the benefit even worth to try? So,
Thank you very much. This is the physician side. Let me ask you a question from a patient side here. If I'm on hydroxyurea, but my hematocrit is not low enough, I get a sangria. Well, I call it sangria here. It's, well, you know, you have a different idea about sangria. But even a kosher sangria. So, you know, you get phlebotomy, and by that I get my hematocrit down. At least this is what they did to me. And I was very happy about it, because I didn't have to take any poison in my body. This is a part of combination, which is, you know, not so sophisticated as you're saying, but we're doing here also a sort of combination. Now, what, for example, if you know you have a patient is on, well, let's put on hydrea, but it's not getting down his spleen, or is on interferon, might be a better example. He's on interferon, but his interferon is not reacting very much in spleen. His spleen is very big. It's not possible, but he takes during a certain period some ruxolitinib, because ruxo, I mean, I tested it on myself. I changed into Jakavi because my spleen was 23 or 24. I still have a belly, but it was about double like this. And it worked perfectly. I mean, Jakavi, after 1 week, all the pressure which I had was gone, and now my spleen is 50% down. Would you agree, for example, if you have a patient like that, he reacts very well to interferon, but his spleen is not reacting on it? So would you give him, during a certain time, Jakavi until the spleen is down? Good question, no?
I mean, I think it depends on a particular patient, but I don't look at it at ruxolitinib as a temporary medication typically. So I think if you add it on, get the spleen down, and then you stop it, I think the chances are very high that it's going to go back up. So I think if you're adding it on, you're keeping it on unless there is a really good reason why you should stop. And so perhaps if you then end up on a combination of interferon and ruxolitinib, which I don't typically do, you know, off study, but I guess I could see a situation where it may be reasonable, then you probably just need to be on a lower dose of interferon and ruxolitinib, but remain on both drugs. Because likely you will get sicker if you stop it.
But as a physician, you could take that decision where you say, okay, you know, this patient is not reacting on the spleen. I help him, whether on short term or long term, I help him with Jakavi.
I think most likely my approach would be to stop the interferon and try ruxolitinib. At least as see what happens. Try that approach as opposed to combining two drugs. And then consider it, but I think that would be the first approach. What would be yours?
well, I would wish to continue both of them if the insurance improves and pays. That's a big problem we have in the US. I have few patients that have been on either Pegasus or Besremi, more likely Pegasus because that's considered off-label in the United States. And then we needed to do Jakafi for whatever reason, And I should not be saying this, but they told me, hey, doc, I have some Pegasus. Should I continue with this? You can guess what I told them.
Yeah,
Yeah, so I concur with what Marina said in terms of the medical side. I'd be nervous to do that. These are drugs that haven't been studied well together, and I'd be concerned about causing harm, which is what we're supposed not to do, first of all. But then I just, this is a global meeting, so just on a more prosaic level, in Israel, I would not be able to do that. The insurance wouldn't cover it. The Pegasus is off-label. We now have Besremi, which is paid for by insurance. It's, it's registered for treatment of polycythemia vera. It's paid for by insurance. But they would not approve both together. So it would be absolutely impossible to do unless somebody was paying out of pocket themselves. And I couldn't do the same with Pegasus either. So technically, it would be literally impossible for me to do that.
So basically, we are dominated by the insurance companies.
Well, from that point of view, yes, but from the other point of view, it's still a combination which is an unknown quantity. And I personally would be a little hesitant.
Okay.
Yeah, in that situation, the safety really comes in there, and we, as mentioned, would have to decrease the dose of one or both of the medications. So we could have ramped up the Jakafi for a little bit if we had an option of both to control the spleen, but then we would definitely run into safety. So we will have to compromise one or the other. So with that situation, if we have no data to back up the decision, it will really be just experimenting with them. That's not what we do. So in those situations, because we don't have any studies, it could be only done on the clinical trial where we have the control of it and we are not causing any harm. But from a clinical situation, it would be extremely rare if anybody's done it just this way.
It's not being done, I know.
It's not, well, the Besremi plus Jakafi has not been done, but it's been proposed that the ruxolitinib plus Jakafi has been done and actually showed quite interesting results and very nice safety. So that has been done. But so far, there hasn't been so much interest from the clinical, from the pharmacy development to combine them, because it's twice the price of the drugs for what benefit. That's what I alluded to at the beginning. We, at that point, even didn't talk about the financial part, but it's twice as many money in it. And then what are we gaining? If we are changing the trajectory of the disease, let's say we combine the Jakafi to control the spleen, and we continue the interferon safely, and in 5 years we can stop them because disease is gone. Well, by no means, we will be all happy and jumping into it. But we don't have that evidence, and then continuing both drugs for what, 20 years? Is it feasible? To how many people, how much, and how safety is it going to be? That things are not answered and are very hard to go in, because with the interferon, now with the more emerging data, we trying to hope we'll be able to stop at some point, or at least decrease the dose significantly, so we don't challenge the immune system forever. But with the other drugs add-on, the concept has to be defined what we do in certain situation, and that has not been even answered from single-agent interferon, because there is just some situation or some patient they're lucky enough to stop and be cured, quote unquote, but none of them would be taking, kind of like a combos. Usually when that is a need, that means that the disease is more complex. When it's controlled by one drug to only some extent, and there is still residual spleen or symptoms or something that basically speaks on itself, like this is probably not the easiest MPN to control. So we see the need for more medicines, but contradictory to that, it's how long can we push the both medicine, because it's a living long with the disease. Not like we've used all the combination with acute leukemias, but it's not for 50 years. That's something that we have not even reached and answered from the safety standpoint and longevity standpoint. But one combination we have not even touched base on was JAK inhibitor and, for example, anemia supporting drugs. I know, and I spoke to some people that take erythropoietin injections on top of Jakafi, or we had other ones such as thalidomide or lenalidomide that being explored and being used for anemias. Now we have another JAK inhibitor that is playing in the game, but that's a combination that is being used for quite time because it's very safe. It does have a benefit in controlling the anemia, but at the same time, it doesn't change the disease trajectory at all. So there, we also don't have a defined endpoints what we do.
Well, sure, but I mean the symptoms, which is it was said today that basically the biggest wish of the patients is that, you know, to changes the transmission, etc. I think it's about the same level as the symptoms. You know, the symptoms which we are having, they are really defining our lifestyle, you know, our quality of life. You have seen the two ladies with, you know, yesterday, very, very extreme. But most of the MPN patients, they have to change their job. You know, either for fatigue or either for the itching or either because, you know, spleen, they cannot move, it hurts, etc., you know. So, I mean, the symptom burdens, you know, they are very big. And, you know, I fully understand, you know, all what you're saying. And when I asked the questions, I knew that you have to reply with, but I think it's important that we have this discussion. So it's really the symptoms. You know, we have been not talking very much about symptoms these days. You know, it's but I wanted to bring it up anyway. You know, the symptom burden for an MPN patient is very big, and you know, we had examples about itching. You know, when we've been at the beginning. You know, I know her, and you know, she really suffered tremendously on itching. Still is suffering. She could take obviously Jakavi, and probably it will be gone. But that's the decision of a physician. You know what he wants to do. But you know, really, the quality of life for us is extremely important. You know, but not everybody has the luck which I have. You know, I can do what I like to do despite of my PV. But most people is different. They have to restrict their life because of their PV, ET, or obviously MF, which is then getting worse. This is why I'm insisting so much on the symptoms.
And I completely agree with this, because we face the same issues from the other side, where we have definition of disease control, right? Where we meet certain criteria, the counts are control, but then I can look at the paper, counts look perfect, nothing, no thrombosis, no risk, and patient suffers. Like, you're not in remission. There's no way you can be in remission when you have bad quality of life. And it does have, well, all the symptoms need to be controlled. Well, to what level? What is acceptable, what is not? And yeah, we have medications that fix symptoms to some point, but as unfortunately, and that's as painful as it is, I have to tell my patient, well, I don't have a symptom miracle drug. I don't. Even the Jakafi work in some point, I still have patients that would be intolerant to Jakafi that we've started for symptoms. And then we are dealing with the side effects on top of a drug that I know personally is the best for symptoms. So then we regroup in a couple months and patient comes like, why don't I feel better? It's like, as I told you, I don't have a miracle drug. I wish I did. So, and the same goes with the interferon. It can cause quite a lot of symptoms on the different levels. So that's a very scary part to also choose and pick. And I was very excited seeing the Roxapex study results, where quite a high dose of interferon was tolerated very well with Jakafi. So it looks like they both work together and complemented each other. So that's definitely a path to go. But we all have to, you know, push that development and go in, because that is something we've been talking about for a long time, because it would help both sides. The symptoms control plus maybe changing the disease trajectory, which is what we have to start looking at.
Do you think that the combos were back in first line of investigation, and well, besides of all the new products coming out, but is the industry again pushing for combos? Is it something which you consider to be really important that we come up with these combinations?
It is important to me. I definitely do see some patients really needing combination up front, where I really need more. And also I have lots of people I would like to get rid of the disease because they're super young. So I would love to have a combo right at the beginning. But at this level, I'm not at that evidence that I could do it for them, because I don't know whether they're going to be the lucky ones and really have a disappearance of JAK2 and complete normalization of bone marrow, and we could stop it. And then what is the trade-in? So I think we have to have drugs that are going to be convincing enough to tell us, yes, this is the way to go. Right now, I don't know if we are at the level, but we always push in combination right at the beginning, because we like to be, you know, as I would say, the real research starts when the drug is approved. That's it. You know, there's no research. That's the most important research we all ought to do as a physicians, and we own the patients. They already put enough to be enrolled in the study and get the drug approved. Now it's time for us all to learn and make it better. So yes, I'm completely with the, hey, let's jump in the combination in every disease. Let's try to eradicate as much as possible. But we all carry the no harm burden where we don't want to do it if we don't have the evidence. But somewhere we have to start.
Hopefully, yeah, definitely. Thank you very much. We have here a person in the room who is a person affected by the combination. He works in, it's Thomas, in the backside there, and he works for Morphosis, and Morphosis has been purchased by Novartis. And I assume it's obvious, but they think that his product at Novartis, Yakavi, together, but they have a good results. I don't know how much you can talk, Thomas, about your situation, but let me know, how do you see it? I mean, you're very much involved in MPN. Maybe you give us your point of view.
Yeah, happy to. And bear with me, I'm not a physician, so I think that was all very, very well said already in the front. But maybe just to add a little bit of commentary from us. And I think first of all, I think it's great to see that there is so much progress in research and developing of additional drugs, combinations in the field of MPNs, because I think we need more. I think we all agree on that. And it's great to see that, especially for myelofibrosis, that there are now combinations coming. Because there are so many, symptoms that we cannot control as best as we can with the monotherapy. So I think that's great. So from our perspective, we see that as a new frontier. And with the data that we were able to present at ASH, it's still early data, as you said, right? It's 24-week data. So we'll continue to monitor it and we'll continue to study it. So there will be longer term data. So hopefully we can see the durability that we all like to see and the efficacy with the manageable safety profile. And so now what we are doing is, of course, now really working with the regulatory agencies, FDA, MNA, put our submission packages together so we get a regulatory approval hopefully as soon as we can. And so that could be a new combination available for myelofibrosis patients hopefully soon. And we also think there is a potential to study this in other MPN ET. We have some early data. So hopefully there's more to come. What's on our minds, in addition, is of course now that, as you said, combinations have been around in other areas for a long time, but if for MPN myelofibrosis, it's new. It's going to be new, and will be, it will be probably the first combination that comes out. So we are really thinking about, okay, what do we need to provide in terms of information to physicians, to patients as well, so we can make those right decisions, like when to use the combination for which patient at what time, and also make sure that everybody is aware to make those good choices. So if we have some insights here from you all guys, what you would need for you and your organizations in terms of to be informed, to make those decisions, and the same is then true probably for physicians as well, to kind of, because we need to think about when to give it, to who to give it, and these things. I think that's what's top of mind for us. So when it is approved, that everybody's informed and it can be used in the right way. I hope that's helpful. Yeah, if you have thoughts, we are happy to hear that, even here in the plenum or like one-on-one in the group in the breaks. Yeah.
Thank you very much, Thomas. Yeah. Laura, as a physician who sees so many patients, what do you think about the combos?
Well, in this kind of clinical trial with the combos, we have experience just with clinical trial, because you cannot use outside a clinical trial. And that is not a political issue, not a money issue. It's a safety issue. If we think we should combine interferon and RUX for whatever, we need to ask for a special allowance, permission, a special permission to the pharmacy of our hospital. It's evaluated by a committee, And there we have to present which is the data supporting that combination. And only in those special cases, we are allowed to use combinations. And we have patients receiving interferon and something else, but those are really special cases where the standard therapy didn't work. That is in MPNs. And regarding the clinical trials where we use these combinations, it's true that we need to find the type of patient that needs these more aggressive treatments, because when you combine, you also combine toxicity, and it's what it's been talked up to now. We need to see how we manage that toxicity and if it is worth it, if the benefit we obtain at the end of the road is worth it for that. I find really interesting the combinations because although the objective of the clinical trial, it's improving the spleen size, improving symptoms, I think that we all want these combinations to change the story of these MPNs. And when we see the mechanism of action of these drugs, that they are different, they probably complementary, we really think this could happen. But we really need to have really good clinical trial designs, because what we were talking before, maybe if we go straight for all to start from the first, we could lose good drugs in this way. So we need to, for example, this CAR-T trial combination that is going to start, that first they receive monotherapy, and then if they don't respond, they go to the combination. I think that's a very wise way to do this, because not everybody needs so much treatment since the first day. So it's a really good news that we have these new drugs, these combinations, but we really need to think for who these combinations are going to be. Because if it's not going to be for everyone, not because of the money cost, because of the toxicity and the benefits that we can get out of this. All these MPNs are not one disease, are patients with MPNs, and they are all different, and the biology of this disease in each patient works different. So they don't need all the same treatment. And I think we need to focus this, because if some treatment works for her, probably won't work for me. So I can go to my doctor and say, I want the same pills she's having, because it's not going to, probably won't work the same way. So I think we have to be conscious on these differences.
Thank you, Laura. Thank you. Now, from the audience, from the patient side, anybody wants to have some comment on that?
Can I have a question? So my question is, isn't there a contradiction between the short-term endpoints and the disease modification that we're talking about? You're talking about that maybe some of these combinations may be long-term disease-modifying combinations, but we stop them because short-term endpoints in the trials, like spleen, or even some of the symptoms or the burden. There's a lot of short-term in these trials. Still, I heard that they might have some long-term disease modification, but we're just losing that possibility. Okay.
Yeah, I think that's one of the frustrating things that the physicians in the last 10 minutes have been speaking about is trial endpoints. How appropriate are they? How appropriate is the timing of endpoints? Who says that a 35% reduction in spleen size is what we should be aiming for? And if it was only a 30% reduction, does that mean the drug's not active? It didn't meet the study endpoints, and if the symptoms improved by 45% and not by 50%. So there's a lot of rethinking that needs to be done in terms of these the endpoints that are chosen, the times at which they are chosen to be measured, and we need need biological data. We need to see that things are changing. And my own personal opinion, and this is sort of taking it a step further even, is I would very like, as a clinician, I would like to see drugs that change the appearance of the bone marrow. Right? The organ that is diseased in MPNs is the bone marrow, and we look at blood and we look at other surrogates. I acknowledge the allele burden and I acknowledge the genomic aspects, but there's an organ in the body that is ill, and it would be great if we had drugs that could actually make that organ work better. I think that would be where I would like to see new drug development going. I think that's perhaps the last stage, because that's a tough thing to do, to change the morphology of the bone marrow. But certainly, I mean, we may have lost drugs along the way because we had the wrong endpoints in clinical studies. And also we had the wrong patient inclusion a lot of the time as well. We haven't developed the idea that all these clinical trials are dealing with patients usually with intermediate 2 and high-risk myelofibrosis, sometimes intermediate 1 you can sneak in. But are we not starting off too late with treating myelofibrosis? Should we not be looking at earlier stages of the disease where the disease might be more modifiable? Have we not lost a lot because we're treating patients that are much sicker and the disease has taken hold of the bone marrow? And it's a much harder thing to do to treat someone who's that ill. I mean, in other examples, in cancer and in medicine in general, we would never do that. We'd never wait for a breast tumor to be so big that it can't be excised, and then, okay, now you can get radiation. So that's sort of, we have a reverse way of thinking about it.
Yeah, that's exactly one of the questions I have. Is there, if you look at the history of the drug development, a lot of attention has been put to MF because of the severity of the disease. But from another standpoint, wouldn't it make more sense to put more energy in the research of the lower risk MPNs to prevent them to go to MF?
I think it would make a lot of sense. But one of the problems with drug development is that we want to have fast results. And so when you take diseases like PV or ET and you want to test the drugs, in addition to just controlling the blood count, which is what a lot of the endpoints are in the trials, what we need to be able to show is lack of thrombotic event and no progression. And that takes years, decades, potentially, and hundreds of patients. Hundreds, right? So this is really difficult to do. This is where we need multiple centers in all over the world participating and to be able to keep patients on studies for many, many years, right? And this is very difficult to do because we don't really have the discipline to do that, or the funding, or all of those. So I think this is a very, very important question, and that's what we should be looking at, but this is why it's not being looked at as much.
Yeah, exactly, well said. And I think one particular point there is safety. Because now we even have drugs for ET-PV that could develop, but even low-grade side effects long-term are not acceptable. Like my patient with PV was refractory to hydria, but could possibly take another drug that is coming in a development. Not like being vomiting and being nauseated for 20 years, even though the drug could change the disease. So once we have drugs that be safe enough for disease that you live with for 30 years, we'll hopefully move on there. But right now we don't. So it's a little bit of a different concept having like early breast cancer where we treat it for 3, 4, 5 years and then we stop. And that we're gonna prevent the disease from progressing. But here, we don't even have safe enough and well enough drug to do something in 5 years. So we have to keep going. That's it, why we decide to start therapy. We tend to tell patients, hey, look, we start. But you have to keep in mind, we'll keep going for as long as it's work. We won't stop it because we don't know whether we could or we don't have any markers. And that goes in a biomarker. Yeah, we may be getting rid of a lot of drugs on the way because we needed some endpoints that are reasonably early but could show us some results, and they were brought up with development of JAK inhibitors, but they surely are not fit for combos because combos are not going to be shown results in 24 months or weeks or whatever we take. It's going to take years for us to see change. That's why a lot of those anti-fibrotic drugs failed. We haven't seen any changes in fibrosis in 6 months and we gave up. Like, really? That's not what we're going to see, a change in bone marrow fibrosis that's been developing for years. And then we give a drug for 6 months and then we don't see results. The company says, well, look, there's no results, I'm quitting, too much money, it's not worth it. That's what we all face the same thing where, yeah, but who's going to commit 20 years with my research to trace the bone marrow and see the results, even though it's something satisfactory? It's very challenging, but we try to move the needle earlier and earlier, but we're not there yet. All the studies, it's a big for us from FDA till we really show safety. We cannot move anywhere else, and that could be only done in refractory disease. That's actually is mandated. We could not even propose study early because FDA would tell us, no, this is too much. You're risking patients' lives, you may harm them, we cannot do this. So we are pushed by the regulators to first show the safety in people that don't actually have any reasonable options, which goes in a refractory disease, and they may be too late. We may have a great drug, but if we test it in somebody who is too sick, we don't even have time to show the results. So there we are facing another issues, how to pick the right patient that is sick enough but not sick enough, that will benefit and not. So that's all of us designing these studies are challenging the same things, and then we propose it to the FDA, and then we get the red light. Like, no, this is not acceptable yet. And then we have to redesign and move a little bit more farther, but that takes us all back 2 steps.
Yeah, absolutely. And so perhaps other ways around this, and something that I think about, and I don't know if anyone's developing this is to, I mentioned it yesterday, to go for things which are much easier to access. And we heard a great talk this morning about metabolism, and I think the theme of inflammation in the bone marrow and systemically has been spoken about a lot in the last couple of days. So what about using simple anti-inflammatory drugs as part of the combo? I mean, and the drug that I've stuck in my mind is colchicine, which is a really harmless drug. It costs pennies, and I would like that kind of thing to be investigated and developed. Yeah, and I mean, we can use simple things, and if the scientists are correct, which we believe that they are, that inflammation is so important in this crosstalk between the malignant cells and their surrounding support cells, then it would be a very easy thing to do clinically, because the drugs are there, they're cheap, they don't need anyone to approve them. You can prescribe colchicine without telling anyone.
Just to add a quick thing to Martin said, this colchicine, we are doing preclinical. Sorry, thank you. Preclinical models we are testing together with Hans Hasselbach. So he came up with the idea colchicine actually. So we are in our models, Jakavi and then also Ropeg interferon alone in combination with colchicine we are testing in our models of myelofibrosis, Martin. Maybe I can give you in the next time when we meet those insights. Mm-hmm. But it's definitely a very good idea.
I'm pleased. I'm pleased that I have the same idea that Hans had. I hadn't discussed that with him, but it seems like an obvious thing to do. And it's borrowed from cardiovascular medicine.
Absolutely. Actually, we got the idea from that. Yes. Mm-hmm. Thank you.
You told us a lot about efficacy of combo therapies in comparison with monotherapies. What about drug resistance? Are combos better than monotherapies, or is there no advantage, or the observation time too short?
That's a $1 million question. They should be. That's the reason they're being combined and done for all other cancer diagnoses where we hit the cancer cell from different angles that prove in CLL and other diseases, they're more complex. We'll just need to learn, as people said, and Thomas and everybody, the follow-up of those studies that use combinations in a prospective fashion. and compare it to the single agent to see whether that's even more durable. Whatever responses we set up, we need to see. One thing is the refractority to Jakafi, because it's a little bit of a different concept than other drugs. The refractority doesn't happen in developing a mutation, like a point mutation in a cancer cell, rather than in a different fashion and changing of the dimerization of the receptor. So that is a big challenge to even define who's resistant. And we face that first with the definition of the suboptimal. Who is suboptimal, who is not optimal, and pretty much what we came up with was, there's no response, but there's no failure. So we kind of get, but still there was not a concept across. And it could come in different flavors. A little bit of a spleen, a little bit of a symptoms, a little bit of anemia, a little bit of a blast. So those are all very likely being driven by different mechanisms that nobody's really looked at and said, this cytokine profile correlates with residual spleen on Jakafi versus this cytokine profiles with residual symptoms. But we still applied the same drug on the combo, hoping it's going to hit it equally. I think that's wrong. And the same thing goes with JAK2-driven versus calreticulin-driven versus we don't know what-driven disease. Though maybe not exactly the same diseases. We, at the current state, lump them together because that's what we've been doing and that's what we know how to define them. But with the genomics coming in, we're starting to learn that one mutation is not the same as two mutations, not the same as three mutations, big role of epigenomes, that's all gonna define the refractority. Perfect example, for example, has been systemic mastocytosis. It's a little bit different disease, but that belongs to the same with the MPNs. And there is a specific mutation that's called KIT that people develop, and then they have a disease. And then over the years, we develop a KIT inhibitors. And there we've seen the resistance patterns. One comes from developing mutation to the drug, the other one from other parts of the disease growing through it. So that was telling there's different flavors of these diseases, and they could progress or get refractory in a different ways. And not until we understand the mechanism which is defining the disease, we're probably just going to be all talking about it. It's not working properly, but we don't know what's wrong with it. Right? That's the best I can answer that question at this level. Maybe Marina has different.
No, I agree. I mean, I think it's just not. Combo should be better than single agent for most patients. Right, theoretically, combination should be better, but as long as because that's how we treat cancers, that's how we often treat like complex infections where we're trying to, fight like tuberculosis. We treat it with multiple antibiotics at the same time to make sure there's no resistance. And it's the same with multiple malignancies. But this is a slightly different disease. It's more chronic, and the pathogen, like how it develops is different. And so we probably need combos, but which combos and how to best, which drugs to combine and at what time points, I think this is the data that we still need. And is it better to do it all together? Is it better to add on the second drug later on? And I think it's going to depend on individual patients and individual drugs. So we're working on this, and I think we're going to have a lot more information on the combination in the next few years with a lot of different drugs. And maybe if multiple drugs that are currently in later phase trials actually show successful results, then we will have different drugs to target to different patients for myelofibrosis.
Just one thing I think important would be for all of us to learn what actually does resistant mean. You asked the question, did you have something specific in mind? I don't know either. It's 40% in reduction of spleen and symptoms enough, or as Peter said, it has to be 100% down. I think it should be as minimal effect in the life as possible for as long as possible. But I don't even know what that means at this level.
We've made up our own definitions, and
Yes.
then we get tied up around endpoints that we can't meet and drugs that don't get developed further. It's a real pity. How about azacitidine as a combination? Did you published on that, if I'm not mistaken? Has that developed in any way?
Yes, that's a fantastic point, which I haven't even talked about. Yeah, we did. Azacitidine is it's a molecule that's been used for quite years for patients with different disease called myelodysplastic syndrome, where the blood cells are coming in a little bit different fashion. I like to say they don't look alike, so they just don't function. And it's an epigenetic model, so it should prevent the resistance, that's the right question, and it should change the environment of the bone marrow slowly. It's not a chemotherapy, it's an epigenetic modulator, or what we call a hypomethylating agents. And we have combined azacitidine and ruxolitinib in newly diagnosed patients with myelofibrosis. Published the results. Actually, we will have an update at ASCO as a poster with one of our fellows, which is going to have a follow-up of about 10 years now, so quite long. And there we have seen responses even at the level of bone marrow. We've seen faster responses in controlling of the spleen and symptoms. I had even patients that had improvements in the counts. So we don't commonly see improvements in platelets. We have drugs right now, JAK inhibitor, that is being used in patients with low platelets. But even there, we've seen maybe 10% people improving the platelets actually. With this combo, I have a patient that started with platelets in the 20s, and now they are in 120, never needed transfusion, which is completely out of the blue. And I had lady that takes the combination now for like 9 years. She calls me once a month at clinic, hey, give me a prescription, I'm going. Which was disease that was very aggressive at start. But it was not seen in like 80% of the patients or 60% of the patient. And then the azacitidine has been an IV drug. So people have to come to clinic every month, and it's burdensome over years. So I'm actually, we're working with, now they are oral compounds that exist as an alternatives and already approved. So hopefully we'll be lucky to work with the pharma companies and propose the oral-only regimen as a combination that's being currently developed in our institution for MDS. We're hoping to do something similar for MPNs, because there would definitely be a way to go with the both oral drugs that could go, and with definitely the intention of changing the course of the disease, particularly addressing the epigenome and the resistance and the evolution of the disease. There's definitely very deep rational, a lot of preclinical, even clinical data on different ones. But this is something that hopefully will build future. But again, here we have to come with something that's doable. Because if it's 5 years, who's going to come to clinic every month for 5 years? Nobody, right? So if it's pill, well, we might get the deal.
Are these trials also in Europe, or is it just in the States?
Well, first, the way it is, we have to show safety. So we usually design it at our place with a smaller group of patients. Once we have the safety, like phase 1 approved with the FDA, then we can reach out to investigators across and do them as a multi-global studies, because at that point the FDA will not be concerned with doing any harm. But usually what it has to start, it starts in a small groups with the safety, and it's very complicated across the continents because the rules are very tight. And again, we don't want to kill the drug before we can see the efficacy. And FDA is extremely careful on the safety signals. If there's 1 or 2, they freeze everything and there's no more development. So there we kind of learned a lesson. And with all of these drugs, we really start small steps. One institution, sometimes we don't even collaborate in between the US, so we know how we assess the study, what research nurses are making the follow-up, how our patients follow. I personally call them and make sure they are right. So, and that is a little slower, but that ensures that we are not harming anyone. And then once we have the signal and we see there is no safety signals whatsoever, then we usually propose amendment and study that could be global. So I'm hoping that will definitely be the movements, because that's something in the future we have in our pipeline to work with companies. But that would be one way to go.
Because I'm sure a lot of even the patients here would love to kind of try it out.
Sure.
It's but also it's a question, how can we as patients help you in all these things? Is there anything which you would need from our side? I don't know. I know information is the most important, but as you can see, what we're trying to do is to try really to make a larger group with more larger information about quality of life and other aspects. So keep it in mind, and whenever you need something, come to us. We are ready to help you there. And John said something before. I just would like to say also, it would be extremely good for us if at least we could have a chronification of the illness of PV and ET, because that will avoid, that problem which we're having, we all in our head have, we might go into MF. And, we know what happens to a few of the patients of MF, and we just don't want to hear about MF. We don't talk about it, but it's here. It's in your head. You manage, the industry manage in CML. CML, you have two medicines, I think now, which is chronifying the illness. Illness is still there, which is, when you have your allele burden at 0.01, but you still have the illness. But, they manage the CML to chronify it. And this is really my first wish on my Santa Claus list. It's really a chronification of PV and ET.
I do really hope we'll get there. I don't know when, but I do hope. It's a perfect example because Michael Keating, Dr. Keating, who's been the greatest researchers for CLL, it's one of the CLL giants, has developed a heavy chemotherapy regimen for CLL back before the 2000s. And that's been a breakthrough. Before, patients were dying of CLL, but that was toxic. So when I met him, which was a complete honor because he's a well-established researcher in his 70s, and I have a dream. I wish we have a pill for CLL as we have now for CML. And then I do have patients with CML that were treated with interferon before the invention of the oral pills that chronified the disease. And that was all that was needed. And at that time, there was this discussion as well that the disease-defining molecule, the BCR-ABL, was chicken or the egg. And there were arguments on both sides saying, it doesn't matter if you control it, the disease is always going to progress. Versus, no, we have to get rid of this, and then it's not going to progress. Well, the other side turned to be correct. We got rid of the BCR-ABL, we chronified the disease, and versus everybody progressing by 3 years to acute leukemias, now nobody progresses. 99% of our patients are alive for 50 years, as everybody else's will be. So the dream of Michael Keating was to have a pill for CLL. Now we have a pill for CLL, and I have personal patient I treated, I stopped treatment, no CLL for 5 years. They should be cured. So we enter the era. So I'm hoping, as long as I live, I will see that getting a pill for MF, and it's not going to come back.
Do you think I can hope it? I'm a bit more advanced than you. Yeah.
I fully agree with you. I think that's one of the things that I was saying earlier, and it really bothers me that we wait for late-stage disease to develop, we wait for transformation to occur, and then we jump on the patients. I just wish there was a way we could move things earlier, and we were talking about even trying to, one of the problems when you diagnose an MPN, the bone marrow is already affected, and it's just so difficult to deal with that. It'd be really great if we had some kind of early detection screening that one day maybe that would really be the answer. I don't know, maybe everyone should have their CHIP mutations tested every 5 years. Maybe that's the dream that we have to go after.
Well, I mean, you have cases, for example, in Basremi. We have actually one of the PV patient who founded with me together MP in Spain. He was on a trial of Basremi, and the trial was finished. He had at the beginning a allele burden of over 90%. It came down to below 1%. And they stopped the trial, and the hospital here, they told him, okay, we want to keep you under control, but we don't give you medicine. It's now 4 years ago. He doesn't take any medicine, and it's still below 1% his allele burden. I call that chronification. Probably it's not. It's not according to your rules, probably not. But this is something. Is it so difficult to find out? Why is it for that person, giving a chronification, like I call it? Or I always thought when something like this has happened, we should be able to find out why is it happening to that person. So, what do we have to do in order it happens to all of them? Or 90% is fine with me, to 90% of the PV patients.
Yeah, it would be great if we knew it. It might be something that's very intrinsic to that individual, part of his immune system, that that couldn't necessarily be copied for someone else. But certainly the example of pegylated interferon for polycythemia, I think that's a little bit of the start of the dream.
Well, that dream I have 10 patients. They are now on our treatment. They have no PV on Pegasus.
Oh, yeah?
Yes. Yes, they've been treating 10 years at least on average. But we stopped therapy, and they even come for bone marrow out of like exercise. Nothing. No JAK2, morphologic normal counts, happy life. And it's 10% of them that we have on the study. But 10% is more than nobody. So, and then plenty others have significant decline in the JAK2 burden, which we were lucky on the trial. We were able to check it every 6 to 12 months, including bone marrows. These people were brave enough to come every year and just do it for the purpose of the science and for knowing. But 10%, I wish it's 80%.
That's a good goal.
And to be even more provocative, I mean, in the IRIS trial, which compared interferon to the first tyrosine kinase inhibitor for CML, which was imatinib, there were 3% of patients on the interferon arm who went into complete remission from interferon alone. So, and that's in CML. So it can do it to some people.
You see? We slow start, but we get big. Yeah. Thomas.
Talking about the combination therapies, do you expect the other JAK inhibitors like... Talking about the combination therapies, do you expect the other JAK inhibitors like fedratinib and momelotinib to work in a similar way with, for example, PELA, or do if can so that you can take the research that you do now with Jakavi and PELA for the other JAK inhibitors, or do you have to start completely new studies and look on every combination as itself, for itself?
That's an excellent question, and we have to start all over again. And every single drug is different and has a different safety profile, so we cannot extrapolate the results from different JAK inhibitors and the combination. I don't know, there we have to really keep up with the side effects profile, where we know that Jakafi is quite myelosuppressive. One of the reasons we were trying combos with Jakafi or ruxolitinib were because of the anemia we're trying to overcome. Fedratinib has quite very similar safety profile, so there are, I don't know, I have some people on fedratinib that then take erythropoietin or thalidomide, they try, they progress. More potential I see with the other two. They are non-myelosuppressive, which is momelotinib and the pacritinib one. There are combinations already being designed and ongoing and starting. We're working on two on our place as well, so I'm hoping that's going to be spread more because those are different patients' populations and different endpoints. And to me, they would be really refractory, more complicated diseases. So with the Jakafi, a lot of the reasons where people develop anemias, which could have been just intolerance or side effects of the drug, and then we combined. But with these two drugs, we will really have a refractory populations of patients, and we will learn a lot about what the combo could do. So we'll have to start again, but we're already working on it, and we all hope to see more combinations with these other JAK inhibitors to be coming very soon into clinical trials and hopefully in a practice as well. Right now, we just combine them what we have. Momelotinib, I haven't seen any combinations with that except for steroids. Pacritinib, I have patients on pacritinib and azacitidine that have overlapping MDS and MPN syndrome, so I could justify using azacitidine, and they had on pacritinib for the myelofibrosis, because I'm treating both diseases at the same time. And there were already some reports by other investigators combining pacritinib, which is approved for very low platelets, with azacitidine. That is something very promising, especially when the disease is already turning more in an anaplastic phase, where we have not much. But that is definitely a way to go, because of these other JAK inhibitors have different properties. They inhibit different pathways that the other ones, more strong JAK1, JAK2 inhibitors don't even touch, especially, for example, IRAK1. It's one of those that pacritinib covers, and that has a huge role in the inflammasome, that we heard about these 2 days. So they could give us a lot of benefits. The problem there is safety. They are a little bit more hard to take, so nobody would like to combine 2 of the same drugs that would really give miserable quality of life. So there we really have to start with the sickest of the sick, but not really too sick, so we can prove it's doable and then move it a little bit more upfront.
I agree with this. I do think that if, for example, pelabresib gets approved or one of the other drugs gets approved, I think slowly we will start combining them with the other JAK inhibitors off clinical trials in the sort of right patient population. For example, like a patient is on momelotinib, and for some reason we believe that they need an additional drug, that perhaps we will combine it. But I think, so as we get more experience with these drugs and as other drugs get approved, I think we will start combining them. But to really get the data on sort of larger scale, we would need the clinical trials.